Welcome, everyone. Thank you for joining us for today's webinar, the current state-of-the-art tissue expression analysis platform for detection and quantification of target and biomarker mRNAs, splice variants, and mutations in preclinical and clinical tissues. It is presented by Christopher Bunker, PhD, Vice President of Business Development at Advanced Cell Diagnostics. My name is Judy O'Rourke, and I will be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Advanced Cell Diagnostics, a brand of biotechny. For more information about our sponsor, visit acdbio.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Bunker. I will now turn the presentation over to him.
Thank you, Judy, and thank you all of you who've joined for this webinar. I'm happy to have a chance to describe to you our RNAscope technology, again, developed and commercialized by Advanced Cell Diagnostics and a brand of biotechny. So it is the current state of the art for in situ hybridization-based expression analysis in fixed tissues. And the areas that I'll speak about as illustrations of that are in the areas of oncology biomarker development and applications in the areas of cell and gene therapy, particularly in relation to biodistribution analysis, again, in the context of intact fixed tissues. So in the 8 years since RNAscope technology was first commercialized, it's seen a tremendous rate of adoption. In that time, we've gone from our first publication in 2012 or 2011, and now we are over 1,800, almost 1,900 publications, with increasing publications at a rate of almost 2 per day. We have over 18,000 RNAscope in situ hybridization probes in use. We make probes to virtually any sequence from any species. They all work under identical conditions, and it's proven to be an extremely valuable way in a tremendous diversity of research areas for doing tissue-based gene expression analysis. The method is oligonucleotide-based. The main features of the assay are that it can be performed on routine FFPE tissues. So unlike historical conventional methods for doing in situ hybridization, which are only really suitable for use on frozen tissues, this method was developed with the primary purpose of enabling RNA in situ hybridization on archival human biopsies with the eventual intention of developing diagnostics. The readout is actually detection of individual single molecules of RNA, which can be quantified on a per cell basis. And because it is oligonucleotide based, it is very reliable and extremely specific and predictably specific based on oligonucleotide melting temperatures. So to give you this is basically workflow of the assay, again, starting with routine FFPE tissues. Archival tissues 5-10 years old have been used routinely with the method, as well as freshly collected clinical biopsies and animal tissues from any number of species. All of the assays rely on paired oligonucleotides for signal amplification. So shown here in the second frame is an example of a pair of oligos where the 3' ends hybridize a sequence specifically to the target RNA, and the 5' ends are shown in a pairwise manner of functioning to hybridize to the rest of an amplification module, which is shown in the middle panel. This allows for the localization of thousands of copies of label, whether they're it's HRP or alkaline phosphatase or fluorophores, all localized to a single transcript, which results in enough signal to detect individual molecules on a per cell basis in tissue. These assays run, we do have manual assays which are used extensively in academic research. Our automated assays run on both Ventana Discovery Ultra and Leica Bond RX instrumentation. And those assays are, again, fully automated from start to finish, deparaffinization to chromogenic detection or fluorescence detection. So fully automated assay on any of these platforms, and I'm going to now illustrate a few of those examples. But first, a few more details about the probes. Again, they're oligonucleotides. No special requirements for preparation of samples. Routine FFP tissues are what these are intended to work on. As shown here, the region that a pair of oligos hybridizes to is about 50 nucleotides. Each oligo hybridizing to about 25 nucleotides. The top, again, functioning in a pair, will hybridize stably to a pre-amplifier, which can then hybridize to additional oligos for amplification. A typical probe actually consists of a large pool of these oligos, all tested in silico, such that we can be sure before the probe is even made that each one of the oligonucleotides is entirely sequence specific for the intended target. The fact that it's just a few oligos out of this entire set are needed enables us to have an assay which has a very high frequency of single molecule detection, despite the fact that the target sequence, the target RNAs, are typically going to be highly fragmented, potentially bound by proteins, potentially degraded, characteristics of the sample which would typically make it hard to detect RNA in fixed tissue. The fact that we're using very short oligonucleotides and distributing them across a region, again, is a feature which enables us to detect transcripts with very high efficiency in fixed tissue. So this is our default. We typically use a pool of oligonucleotides of this length. It could be longer or shorter depending on the target sequence. And just to kind of walk through how this works again in terms of sequence-specific binding, amplification through sequential hybridization steps, where we're hybridizing one oligo to the next, and then ultimately coming in with either chromogenic enzymes or fluorophores as the label, much like you would do with IHC or immunofluorescence. But in this case, again, rather than requiring thousands of antibodies to bind to thousands of protein epitopes, we're localizing all that signal to individual transcripts. And the other benefit of the assay is that the signal amplification is dependent on multiple things happening simultaneously. And as a result, there is essentially no background because it's virtually impossible for pairs of oligos to hybridize right next to each other in a random manner. As a result, it's a very, very clean assay, which allows us to quantify these signals as digital measures of expression in tissue. This is just I'll speak very briefly to this slide here. This is one of the first publications in which a comparison between RNAscope, shown on the left-hand side, and the green signal you see scattered around the nucleus is HER2 transcript, and the panel, the graph on the right-hand side is showing a solution-based quantitative measure of HER2 transcript from HeLa cells. So both are HeLa cell samples. One is we're quantifying or measuring HER2 expression by RNAscope, and on the right-hand side we're showing a quantification in solution. And what you see, the circled numbers show we're getting very similar numbers, whether we count dots visually or whether we use a solution-based assay and a standard curve of in vitro transcribed message. And this points to the fact that when we see dots, the dots represent actual individual transcripts, which we can quantify using various softwares. The software that we typically use is called Halo from Indica Labs. Other softwares from Definiens, Visiopharm, Cell Profiler, which is free software, all of those can perform the same function. And these are softwares which are designed to identify spherical round objects as dots and then quantify those, count those on a per cell basis. And so we can count individual dots. We can count dots based on different markers and different colors to get measures of both expression as well as percent cell positive, percent cell dual positive. For instance, even three. We can look at three markers simultaneously. So we I use this this example, or this example data shown on this slide, to kind of compare and contrast with a sort of the gold standard for molecular-based quantitative analysis, NanoStrin, an extremely sensitive assay, which does actually work on formalin-fixed tissue, and which also has a very wide dynamic range. What's shown here is the fact that we're looking at TNF-alpha, IL-1 beta, and CTLA-4 in non-small cell lung cancer tumors. The numbers at the top, average dots per cell being less than 1 per cell, and whole tissue NanoStrin analysis giving a value of 1 for each one of these markers, meaning expression on a per cell basis is extremely low, almost undetectable. However, what's obvious from a visual analysis for any one of these markers is you can see a very few cells expressing it at quite high levels, particularly in the case of IL-1 beta. And that's the value of a quantitative assay like RNAscope, which also gives you a visual means of looking at expression on a cell-by-cell basis. So in that way, it's been used quite extensively to validate or corroborate transcriptomic type approaches where you can get information from an entire tissue or cell population across the entire transcriptome, and then use RNAscope to assess whether it's specific cells in that population are expressing, or whether it's more uniform expression throughout the entire tissue. So the fact that we can detect a very few dots corresponding to very few transcripts turns out to be very, very powerful and very relevant information about targets and biomarkers. And that's illustrated in the next 2 slides. This one is showing data from Bluebird Bio, which is developing CAR T-cell therapies. And this is in relation to RR1, or receptor tyrosine kinase, which has been considered a candidate for both ADC as well as CAR T therapeutic development. And it's this data, the data on this slide, which the primary piece of data is in the center here. And this is showing very low expression, low-level expression of RR1 in mouse pulmonary endothelial cells. And this expression is entirely undetectable by in situ hybridization. Excuse me, IHC. But it is detectable, obviously, by in situ hybridization. And although it's only a very few dots in a very few cells, this level of expression is sufficient for engagement of CAR T cells that target RR1. So the data shown on the right-hand side, lower right is showing RNAscope CAR T detection. This is showing detection of CAR T cells in mouse lung, and the larger panel showing granzyme B expression is the result of RR1 targeted CAR T cells infiltrating into the lung and attacking these cells and resulting in a granzyme B expression from activated T cells. So this this points to the fact that extremely sensitive detection, in this case, points to the toxicity risk, on target, off tumor toxicity, resulting from low level expression in this case mouse lung. This level of expression it was shown to occur in human lung as well, which is why RR1 is no longer a target for CAR T development by Bluebird Bio. Similarly, this is data from Merrimack Pharmaceuticals, which also is targeting. In this case, they're looking at the expression of heregulin. They have a therapeutic program targeting ERB-B3. MM121 is an anti-ERB-B3 neutralizing antibody in clinical development. And they determined that its expression of heregulin, which is the growth factor ligand of ERB-B3, which is the predictor of patient response. And so here again, we're seeing very low level expression represented by a few dots in this case, non-small cell lung cancer tumor tissue from a patient. It's this level of expression, although it's completely undetectable by IHC, it does predict response. And it was the basis of these sort of results led to the use of this assay as a means of selecting patients for enrollment in later stage clinical trials. So these 2 pieces of data are just examples of where very low level expression can both identify potential adverse events or toxicity if it's expression in normal tissues, or where very low level expression in tumor microenvironment could point to patients who would be responsive but might otherwise be identified as a false negative by a less sensitive assay like IHC. So just to very quickly go through a couple of examples of different kinds of assays that can be performed with RNAscope. These are single marker assays, either using HRP or alkaline phosphatase on a variety of different tumor types. We've recently, we have assays which run on both Leica and Ventana platforms. We've recently developed some new and significantly improved assays running on the Ventana Discovery Ultra instrumentation. And this is the data shown here is from that. This is just tonsil tissue showing some markers that are quite difficult to detect by IHC, namely CXCL9 cytokine and IL-12 cytokine, IL-12 cytokine, and CXCL9 chemokine. Very low level expression can be seen. The red signal for IL-12 is quite low, but visible. And again, in relation to RR1 in mouse lung or heregulin in tumor, that low level expression is likely to be meaningful. Likewise, VISTA and TIGIT expression in populations of cells within, again, within a human tonsil sample. Some of the more advanced assays we've recently developed and have recently commercialized involve both combinations of dual in situ hybridization combined with IHC. And that's shown here, looking at FOXP3 and TGF-beta by in situ hybridization and combining that with CD4 IHC detection, also running on Ventana Discovery Ultra instrumentation. So these are just hopefully more than just pretty pictures, good illustrations of the ease with which we can combine essentially any probes and some antibodies, where we are continuing to work on the validation of different antibodies to be used in conjunction with in situ hybridization probes. And we're mostly focusing on developing IHC assays for cell markers that we'll work with in situ hybridization, as we feel that will be of most value, particularly in the context of immuno-oncology. But again, the fact that we can detect virtually any transcript with RNAscope probes, we have probes for nearly, not all, but the vast majority of relevant targets, particularly in immuno-oncology. And these can be run on essentially any tumor type as well as normal tissues, provide a means of getting a very rapid view, a quick turnaround time, view of expression in any sort of intact tissue. And so, again, to that point, and speaking to the fact that it's used quite extensively, published frequently on in papers describing transcriptomic studies, RNAscope is very frequently used to validate results observed either through transcriptomics, RNA sequencing, gene chip studies, et cetera, because it takes so little time for us to develop probes, just a matter of 2 weeks. They're essentially ready to use as soon as they're made, and data can then be generated in a day. So it becomes a very efficient method, both of evaluating novel targets coming out of transcriptomics, or if there's a very immediate need for biomarker development, the method can be applied very quickly there as well. That was actually the situation with Insight and their IDO1 program. They had been working on an IHC assay for almost 3 years, and then realized that it was not going to be sufficiently sensitive to meet their needs in clinical therapeutic development. And within just a matter of a couple of months, we were able to show them that the RNAscope assay was about 10 times more sensitive than the IHC assay that they'd been working on for so long. And since then, we put this assay to very extensive use to analyze a large cohort of patients to measure IDO1 expression across many, many different tumor types. But there again, a good illustration of where we can develop assays very, very quickly, demonstrate their performance against other methodologies, and most oftentimes we're able to demonstrate an assay which actually has higher sensitivity and oftentimes higher specificity than more conventional IHC-based methods. So now to sort of segue into another area where we're seeing a great deal of use of our technology, and we're really excited to see the contribution that this assay is making in the context of CAR T cell and TCR-T cell therapeutic development. The illustrations here are showing on the left-hand side the one of many lentiviral constructs with a CAR chimeric antigen receptor insert in the middle of it, and on the right-hand side showing the resulting protein. The point here is that the protein made by CAR T cells and TCR T cells, that protein is almost entirely human, or if not entirely human, meaning it's nearly impossible to differentiate a CAR positive T cell from a non-CAR positive T cell in a human patient biopsy. In contrast, the transcript that's making the CAR protein has viral untranslated region sequences at the 5' and 3' end. And these provide unique sequence tags that are only present in virally transduced T cells, CAR T cells, TCR T cells. And this has been put to great use and continues to be adopted more and more widely as a means of tracking CAR T cell activity and CAR T cell tumor infiltration in patient biopsies. So most of these trials now appear to be involving taking or collecting both pre-treatment and post-treatment biopsies from patients to assess tumor infiltration and activity of those CAR T cells or TCR T cells. This is one example from Adaptimmune presented last year at ASCO, and what's shown here is data that ACD generated for them showing the infiltration of TCR T cells. These TCR T cells are targeting the NY-ESO target, and the TCR is shown in red. And again, this is using a probe to a UTR sequence in those T cells. And then associated recruitment of CD3 positive T cells using a greenish blue signal, also an RNAscope readout. So that's one illustration. Another one this is from Juno, actually 2 years ago at a SITC meeting, also showing pre and post treatment diffuse large B-cell lymphoma patients. And here they're simply using a chromogenic DAB readout to detect the presence of their CAR T cells in post-treatment biopsies. I use this data. This is from their poster also showing detection of IDO1 and PDL1 by IC. Personally, I felt those images were not all that impressive and somewhat difficult to interpret. And I use that to kind of illustrate both the ease as well as the very high signal to noise of comparable assays using RNAscope, in this case showing IDO1 and PDL1 detection with an assay which is much cleaner and has higher signal to noise and can be easily multiplexed. I think it becomes a much more valuable and an informative experiment to be able to look at multiple markers, both in this case a CAR transcript, as well as markers that might be related to checkpoint inhibition and T cell inactivation, like IDO1 and PDL1. A final example. Preclinical data. This is again from Bluebird Bio. Give credit to Jim Rotman for collaborating with us on generating these data. Also relating to RR1. Here showing, again, in the first place, showing the very low level expression of RR1 in mouse lung, and the result of treating these mice with CAR T cells that target RR1. And that's shown here. Those in that same lung tissue. What is shown and what is observed are CAR T cells infiltrating into the lung, becoming activated, as illustrated by granzyme B expression, and the resulting phenotype being pulmonary toxicity and respiratory issues. Another consequence of the T cells infiltrating into the lung and becoming activated there is they apparently never actually reach the tumor. This is a xenograft tumor in those same mice. We see the same very low level expression of RR1 across the tumor. It's known to be positive for RR1, but there's no red signal on the left-hand panel. There's some green signal which corresponds to granzyme B, but it appears that the T cells, CAR T cells, do not actually reach the tumor, presumably because they were sequestered or became activated and exhausted in the lung microenvironment. These are studies done on mice. We do just as many studies on rats, other therapeutic areas, non-human primates, partly for safety assessment to look at target expression across normal tissues. We also do studies across normal human tissues, again in relation to target safety assessment. The areas where we do a great deal of work in non-human primate, rat, and mouse is in the area of gene therapy. And here again, much like the situation with CAR T cells and TCR-T cells, where we're using a viral sequence as a means of identifying those cells, here we are detecting the AAV virus. And so the objective of these studies, in the interest of our clients for whom we work, is to look at the biodistribution of their AAV gene therapy vector. We can do that with the RNAscope assay. So despite the fact that it's called RNAscope, AAV DNA genomic sequence is as accessible as is RNA. It's an episomal a DNA sequence not tightly packed in chromatin like the genome of the species. So we can detect AAV just as well as we can a transcript. So shown here, this is sort of low magnification, showing on the right-hand side a part of a non-human primate eye where AAV was administered. On the right-hand side, showing no administration, no signal. The probes we're using are a combination of a probe to a CBA sequence. This is chicken beta albumin promoter sequence, and GFP, a transgene being used to look at expression from that vector. So if you look at higher magnification, what we see is this sort of bluish green signal in some nuclei. That, again, corresponds to detection of the AAV DNA, which is nuclear, and then the red signal, which is predominantly distributed across the cytoplasm outside of the nucleus, and that's, again, with a probe using GFP. So this has also becoming a standard methodology for, as some Pfizer investigators describe it in a review, gene therapy review, morphology-based biodistribution analysis is what this provides, in contrast to taking this tissue, grinding it up, and doing PCR to measure the presence of AAV. This is a very recent paper just published in Nature Medicine. Here again, same story, same application, using RNAscope to measure the presence, and in this case, determining the localization of their AAV vector to photoreceptor cells in a non-human primate eye. In this case, what they're actually delivering is a CRISPR-Cas9 sequence. The Cas9 is being delivered by gene therapy with the intention of gene editing to correct ocular disease. So these assays, everything I've shown you in the previous slides, are all illustrations of the RNAscope methodology. They all work in essentially the same way. This very similar probe designs, whether they're running on animal tissues or human tissues, it's essentially the same process. And again, these assays are all run fully automated on Ventana as well as Leica instrumentation. At ACD, the majority of our business is selling research products, which are used by academic and big pharma companies. We also offer pharma assay services, which serve primarily the biotechnology community, which is not invested in autostainers and other capital equipment like that. And for them, we run thousands and thousands of assays, tens of thousands of slides. And again, with a feature of an assay which requires very little development time, we can turn around results very, very quickly. And what we can provide with this is gene expression, target expression, or viral distribution analysis in a variety of different tissues, oftentimes multiplexed with cell markers, whether it's neuronal cell markers or tumor cell markers, with readouts that correspond to molecular detection, which can be quantified on a per cell basis. And so with that, I will say thank you again for your attention and your time, and I would be glad to take any questions you have.
Thank you, Dr. Bunker, for that informative presentation. I would also like to thank Advanced Cell Diagnostics for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May 27th, 2019. And as a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we'll see you next time. Goodbye.